Hello, I'm Josh Starmer and welcome to Human Stories in AI with StatQuest and Lightning AI. In this series, we'll hear about the career journeys of passionate AI experts. From their humble beginnings to conquered challenges, we'll be inspired by the real-world experiences of professionals thriving in the ever-evolving AI landscape. Human Stories in AI is brought to you by Lightning AI. Code together, prototype, train, and deploy AI web apps, all from your browser with zero setup. Personally, I love Lightning AI because it makes it super easy to use and learn from the StatQuest coding tutorials. Just go to the webpage, click on the Run button, and bam! You get code that you can play with without downloading anything or installing any packages. Today, we have special guest Tommy Tang, the Director of Computational Biology at Immunitas Therapeutics. Tommy is a computational biologist with over 10 years of computational experience and 6 years wet lab experience, committed to reproducible research and open science. At Immunitas Therapeutics, Tommy employs a single-cell sequencing platform to dissect the biology of immune cells in human tumors by using machine learning approaches and cloud computing. Before all of this, Tommy was a lead scientist at the Dana-Farber Cancer Institute. So, without further ado, Tommy, can you tell us about your journey to where you are right now at Immunitas Therapeutics? How did this all start?
It dates back to my childhood back in China. So I'm from China. I was born in China. In the south part of China, it was in a small town. I was born in a low-income family, and my dad was a middle school teacher, and my mom didn't have a job. She was caring for me and my sister, and that was more than a full-time job. And we could never make ends meet. And I remember there was this one day we couldn't afford any meal, and we could only have a soup with soy sauce and green onions. That was really hard. My mom always told me, Tommy, you need to study hard, and only knowledge can change your life. So I studied really, really hard. So I actually got into the best high school. I was ranked number 1 in the county. And then later I got into one of the top universities in China. That's there in Shanghai, and that's where I had the opportunity to learn a little bit more like what's out there in the world, and because it's in a big city, and we also had a really good environment to go abroad. And I applied and many graduate schools in the U.S. So 2008, August 8th, I came to the States at the University of Florida to pursue my PhD at the University of Florida. And I was actually trained in a wet molecular biology, cancer biology lab, and I didn't know any data analysis skills. And at first year of my PhD, my advisor asked me to analyze this public available chip sequencing data set, and it crashed my Excel. That was the first time I realized, okay, no matter how good my hands are in the lab, I need to learn some computational skills. And I started to learn computational biology online, like taking courses on edX, Coursera, and Udacity in the early days. And I just enjoy doing that, because after the busy days in the lab, I dash back to the apartment and then start to do the exercise, do the homework, and also Google a lot online. And kind of learned a little bit computational skills in the last 2 years of my PhD. Then I moved to MD Anderson Cancer Center as a full computational biology postdoc. And there my advisor was Dr. Rova Hack, who was leading the Cancer Genome Atlas glioblastoma project. So I had the opportunity to analyze real large-scale genomic data sets, TB size. Yeah.
Now, that sounds like a really big step of you were a lab biologist and a self-taught computational biologist. This is all stuff you did on your own. I mean, you took some online courses, but no formal degree, no master's nothing like that. And now you've got a postdoc, which is basically a job, full-time job in computational biology.
Correct.
Was that stressful? Was that like, oh, this is just no big deal? I took these online courses. I'm qualified to do this at this huge cancer center. Tell me about that.
Yeah. It was definitely stressful, right? Although, it took me 6 years for my PhD, and the first 4 years, I already had all the publications, but they are all purely wet lab based. So that's why I spent 2 more years, to kind of learn a little bit more, Unix commands, basic R and Python. So I was kind of better prepared for my postdoc, but of course, even after 2 years, I was still a beginner. So I learned quite a bit actually during my postdoc at MD Anderson Cancer Center, and they are also friends, good colleagues in the same lab also helped me a lot, just to learn a little bit more advanced skills. But again, everybody is busy, and even they teach you something, you still need to spend a lot of time just by yourself and do a little more studying after they teach you something. Yeah.
And from MD Anderson, what happened?
Oh yeah, so I stayed there for 3 years for my postdoc, and there I learned quite a bit on different sequencing data analysis, different types of sequencing data, whole exon, whole genome, bulk RNA sequencing, chip sequencing, ATAC sequencing, some reduced bisulfite sequencing to study DNA methylation. So I learned quite a bit of different data types, and then I also learned this workflow language called SnakeMake.
Okay.
So, and because in my later years at MD Anderson, I was in Dr. Kunal Rai's lab, and his lab actually developed this high throughput chip sequencing assay, and we were actually profiling 6 different histone modifications across hundreds of samples, and if you times 6 and plus input control times 7, and that's thousands of samples, and we had to actually process those samples uniformly, and that's how I started to learn SnakeMake and wrote this pipeline to process all this data in the lab.
So to summarize for sort of a general audience, you guys were collecting data, sort of genetics-related data, and you were collecting a lot of it. And what you did is you created a pipeline for processing that data and analyzing that data, and you used it using something called SnakeMake, which sounds fascinating in and of
it's just a Python extension, yeah.
Okay, so anyway, so just to summarize, so that we're all on the same page. Okay, so you've created this cool pipeline for processing large amounts of data.
Yes. Yeah. So then I extended my Snake pipeline to process all kinds of different assays that I've mentioned before. So looks like I have a good tool on my belt. And then I moved to Harvard Faculty of Arts and Science Informatics Division as a senior bioinformatician. There I started to analyze single-cell RNA sequencing and single-cell ATAC sequencing data, so in collaboration with the neuroscience lab. So I stayed there for 1.5 years, and I moved to Dana-Farber Institute to actually lead a big NIH-funded consortium project called CIDC, Cancer Immunological Data Commons. So in that project, 4 different cancer centers in the States, MD Anderson, Dana-Farber, Mount Sinai, and Stanford, they carry out immunotherapy clinical trials, and they profile those patients using next generation sequencing technology like whole exon, whole genome, and not whole genome, TCR sequencing and bulk RNA sequencing. And maybe it's by coincidence they were using SnakeMake. So that was a perfect position for me. So you never know what you learn can be useful.
That's hilarious.
Point.
Yeah.
And in collaboration actually with Ethan Therami's group at Dana-Farber. So their team, they are more like a software engineer team. So we actually process all those clinical trial data securely on the cloud, and then they will actually package our uniformly processed data sets into a really nice web portal and deliver it back to the clinical trial team for them to do downstream analysis. And 2 years there, and I joined Immunitas Therapeutics, and as Director of Computational Biology to establish their computational biology capability. And Immunitas is a single-cell genomics company trying to use single-cell RNA sequencing data and single-cell T-cell sequencing data to find new therapeutic targets for cancer patients. And we also have an antibody engineer team to generate those antibodies. So the wet lab biology team can test those two antibodies and eventually get into the clinic. Yeah.
So again, just to like bring everyone up to speed, what Tommy says when he says single cell, that means that like you can take, so like your body is made up of all types of cells. They can take an individual cell and look and see what's going on within it, rather than say like a whole bulk of cells and just sort of take the average of it. They can look at things on a single individual cell basis. And then it sounds like you guys can then develop potential therapeutics based on those results. So you've got sort of the basic research to identify targets, and then there's a component that develops things that would bind to those targets and hopefully cure cancer or some disease.
Correct. Yes, exactly.
Yeah, well, that is fascinating. Well, I can see why you'd be excited about being there.
Yes, and just to note, like our leading program is called IMT-009. It's a CD161 antibody. So CD161 is a T cell and NK cell modulator. And hopefully, if we can block it, then it can activate both T cells and NK cells and try to benefit the patients. And now it's in clinical trial phase 1. We dosed like 7-8 patients now. It's safe. And I'm really excited to see whether it's going to like benefit like patients. And
that's amazing. So just so I understand, make sure I hear what you're saying, because this sounds fantastic and super exciting. You guys did some exploratory research, some single cell stuff. You came up with a molecule that can then target things that you discovered, and that molecule is now in clinical trials, and it looks like it's working, is what you're saying.
Just to clarify, this specific molecule was actually discovered by our co-founder, but using the same single-cell RNA sequencing, single-cell TCR sequencing platform. And when I joined the company, like this molecule actually was already identified. What I was doing is to actually using the same platform and trying to identify new targets.
Okay. Oh, so that's what you did. Okay, fantastic.
Yes, and also now we have this drug on the clinical trial. We're dosing the patients, and we also generated data from the patients. So we are also supporting the biomarker data analysis from those clinical trial.
So the molecule's there, but what you did is you found some new targets for it.
New targets for like new antibodies to target.
Okay.
Yeah.
Okay.
Yeah.
Fantastic. Well, that's very exciting. Would you mind sharing sort of like what techniques you guys are using? I mean, you've talked a little bit about SnakeMake. But is there any sort of like, do you guys use machine learning at all, or artificial intelligence, or what? How do you interrogate your data?
Yeah, that's a great question. So at Immunitas, we actually have a machine from 10x Genomics to generate single cell RNA sequencing data and single cell T cell receptor sequencing data. So what it can do is that for a single cell, you can have the gene expression profile, but you also know the T cell receptor sequence. And what you can do is, you can correlate the gene expression profile with the clonal expansion or T cell phenotype, because when a T cell recognizes some antigen, it expanded. For example, it could be a tumor antigen. So when a T cell recognizes it, it kind of expanded and try to destroy the tumor cells. And when the T cells expanded, all those same T cells, they will have the same exact TCR sequence.
Okay.
So by just counting how many times that T cell receptor occurs in the cell population, you know, how much expansion that T cell is expanded. And then you can correlate the gene expression profile with the T cell expansion phenotype. And you were asking what kind of machine learning approaches we use. Then we always start with simple, interpretable like machine learning approaches. For example, in this case, you can use we actually use single logistic regression. For example, you can designate whether a T cell is expanded or not expanded. Then you can essentially find correlation or anti-correlation of your gene expression to that phenotype. So either random forest or logistic regression are used for that purpose.
Fantastic. Just out of curiosity, I mean, I'm going to be honest, I'm no immunologist. And so a lot of what you said sort of just went right over my head. But let me see if I can summarize things. So, like when I get a vaccine for something, what happens is, one, I get a sore arm, but then two, I guess there's an immune response that ramps up against the vaccine.
Correct.
And that's that T cell expansion thing. Is that what you're talking about? How my body's making more cells that can attack whatever I'm getting immunized against.
Correct. So I think I'm not an immunologist either, but I learned quite a bit just by working with them.
Oh, I bet, yeah.
And I think because the vaccine, like previously people used those maybe just died with a virus, inactivated virus, and you inject into the body. And the T cells, as you said, they actually recognize them, right? And then they will expand and try to clear them. And then those T cells, to get memory, they call it T memory cells. And so after it clear all those viruses, and they become dominant, but they keep the memory. But next time when the real virus come, and those T cells have memory, so they can recognize it and expand really quickly and get rid of them. Yeah.
Oh, fantastic. And so what you're doing is, so you're checking to see if the T cells have expanded or not, and you can do that just by counting the number of, I guess, T cell sequences that you're getting, and you're correlating that with single cell RNA gene expression.
Correct.
And what cells are those? Is that a different cell type, or is that the same?
Those are the same T cells.
Yeah. Same T cells. Okay, and so when you're like, okay, we've got a bunch of T cells, and they're all we sequenced the receptor thing, so we know that the immune response is kicking in, and we're building a large collection of these T cells. You can then say these genes positively correlated with this transition to expanding and sort of building an immune response.
Yes.
That is very cool.
Yeah, there are many actually nuances under under this. For example, not necessarily. For example, one famous example is PD-1. It's the most famous checkpoint for immunotherapy.
Okay, so tell us about PD-1.
So,
this is all new for me.
So PD-1 is an immune checkpoint on T cells. So when the T cells are activated, so then the T cell, the PD-1 expression will also increase actually. And what the tumor cells can do is that they express its ligand, called PD-L1, PD ligand 1. And the tumor cells will use the PD-L1 to block the PD-1.
Oh, so what they do is, so there's this PD-1 thing.
Yeah.
And does PD-1 promote an immune response?
So yes and no. So PD-1 is more like PD-1 is more like active. So when the T cells expand, PD-1 actually is up, and it's trying to actually. But then when PD-1 is too high, and it's also trying to damp the immune system, because you don't want to over activate the immune system.
Okay.
So
with the tumor has a defense against PD-1, and you're saying it creates something called a ligand.
Yes.
And a ligand is something that sort of fits into like imagine PD-1 is like a glove, and the ligand, the PD-1 ligand that the cancer creates fits into that glove. And what is that? What is the effect of that?
So the effect will be the T cells will actually bypass those cancer cells.
Oh, that's very sneaky.
Yes, that's very sneaky. Yeah,
Is that breaking the rules? I mean, is that fair?
it's not fair. Yeah, but tumor cells come up with those mechanisms. Like they upregulate PD-L1 and then binds to the PD-1 on the T cells, and T cells will actually just will not work on those.
Oh
cancer
wow.
cells.
Okay. So are you looking for ways to bypass that checkpoint, or are you guys trying to figure out a way to prevent the tumor from tricking the T cells?
That's already a drug in the market. Like it's very like well-known drug, and it's anti-PD-1 antibody. So the antibody will actually binds to the PD-1, but only binds to it, but so occupy that pocket. So then the PD-L1 from the tumor cells or from other immune cells, they cannot.
Sort of bounces off.
Yeah, it's already preoccupied by this antibody. So the T cells will remain active and then try to kill the tumor cells.
Okay.
Yeah.
And so that's one of the things you're looking at when you're correlating with, you're doing this logistic regression, or you're doing a random forest,
Right.
and you're saying we're looking at expression of genes, including maybe the expression of PD-1.
Yeah. And in fact, PD-1 show up in our analysis, and that shows, okay, our analysis is making sense because, okay, PD-1 actually is up in those highly expanded T cells. Yes.
So that's sort of a proof of concept. It's a way of calibrating your test to make sure it's actually working correctly.
Yes, yes.
Oh, fantastic.
Yeah.
So that's a way to, is that sort of used to train your random forest and to train your logistic regression and figure out what thresholds to use for classification? Or?
So in this case, we're not using it for classification. We are looking at the weights of the features or the genes. So then we say, okay, which gene is positively correlated or anti-correlated with this T cell expansion?
Okay.
But again, think about it here. Like biology is more complicated. So PD-1 is like positively correlated with T cell expansion. And T cell expansion is a good phenotype we want to have.
Yeah.
Do you want to inhibit PD-1 or do you want to activate PD-1? So in this case, actually, we are inhibiting. We're not trying to activate PD-1.
Okay.
Yeah. So the biology is kind of complicated because the T cells, they upregulate PD-1, like when they are they're expanding. But then the immune system actually also trying to like damp down the immune response because it's a balance within the...
Yeah, so it wants it to grow, but it doesn't want it to take over the entire body. It wants it just to it's a way of promoting something, but then saying, okay, enough.
Yeah, exactly.
I don't need to eat anymore. I've had enough. I've had my fill. Let's start toning things down. And
and also PD-1 is also a like immune like exhaustion marker. So immune exhaustion means the T cells, they are activated for too long.
Okay.
And then PD-1 is high, really high, but then they lost the ability actually to attack the tumor cells, so they're exhausted. So that's why if we want to try to actually block the PD-1, and some people say try to reinvigorate those like exhausted T cells.
Yeah. Well, so it sounds like you've got relatively large data sets. Is that correct? And the question that follows up from that is, how do you guys manage all that data? Do you do it in-house? Do you have like a huge data warehouse on site? Or do you lease space in the cloud? Or how do you manage all that data? And how do you manage your pipelines in terms of where's the computing happening? And
we're using Google Cloud.
Okay.
And so Google Cloud, so the good part of that, you can spin up any size of a virtual machine and with a big enough RAM and disk space. So single cell, you can sometimes have millions of cells in a single data set. It can be huge. So we can't all process the data on our desktop. So we have to use the Google Cloud. But then I told you we sequence those. We actually make libraries in-house, but then we send it to Broad for sequencing, and we grab all the FASTQ files and then send it to one of the virtual machine, and we actually also write SnakeMake pipelines to crunch those data in-house. We have a pipeline for single-cell pre-processing. Yeah.
So once you get the data all in the cloud, you then run this SnakeMake pipeline, data analysis pipeline,
Yes.
To sort of do all the analysis for you, and that's also done up in the cloud.
Correct.
Yeah. Just out of curiosity, how long does a typical analysis take to run?
For pre-processing, it doesn't probably take that long. Also, but that depends on what tools are you using. From 10x Genomics, they have their commercial solution called Cell Ranger. Is a pipeline they developed, so you can run that. But that can take you like tens of hours just to do the pre-processing from the FASTQ files to the final count metrics like rows are genes, columns are the cells. So, and if you use some other tools called like Crystal, Bastool, or like Cellman, those are so-called alignment-free tools, and they also have single-cell version for quantification. And usually that takes like within 1 hour or something like that. It's much faster.
So it's not too bad, yeah.
Yeah, but then the challenge really is for downstream analysis. Also, for example, for cell type annotation, this is more challenging. For example, because single-cell, like, first, the size is big. Second, there are many zeros, so the data matrix is kind of sparse. You have to treat them a little bit specially. And for cell annotation, that's one of the most challenging problem, because after usually people do, they sequence a bunch of cells, for example, in the tumor, but the tumor mass contains so many different cell types, different immune cells, cancer cells, and you want to actually classify them using either like marker-based method or like machine learning approach. But the thing is, no matter like how much work you do try to automatically predict those cell types, if you work with the immunologist, they will always actually change your annotation because...
Oh, for real?
Yeah, because the immune cell state is very complicated. You can't... There's no one-fit-all actually annotation nomenclature, different cell states, and that's one of the big challenges there. Yeah.
So I guess a question I have is, how do they know?
There's no ground truth here.
Yeah. Okay.
That's the hard part. Like, I think for simple data sets, like some people like. They can do some like synthetic like data sets, like more like artificial data sets. For example, they have different cell lines. They know they're kind of homogeneous for each other, but then they mix, for example, 3 different like cell lines together, and then you sequence them. Then you cluster them. Okay, they probably are separated into 3 different clusters. That's kind of very clear. But biology is always more complicated because the cells can have different cell state. From this state to this state, it's a continuous like a process. You cannot like just make a discrete whatever cell type based on the gene expression. It's very tough. Yeah.
So when you're looking at individual cells rather than the average of a big pile of cells,
yes.
When you're looking at individual ones, they may all be from the same cell type. Mm-hmm. But they could be doing different things or a different stage in their life cycle or something like that. And so they may not all, they're not like, I mean, in some sense they're clones, but they're not all doing the same thing. Yes. They're not all like, we're doing the exact same thing. And as a result, it can be tricky. So when they're doing different things, that means they're using different genes and activating different things. So when you're doing the analysis, it can be difficult to say, yes, these 2 cells are the same type. They're just doing different. They're at different stages in their lives.
Yeah. I think exactly you're right. Like some cells, they are of the same cell type per se, but they they're of different state. They're like different. They're actually exact their function a little bit differently, and it's also a little bit philosophical question. Like when you define cell types, you're trying to cluster the cells, right? But then when you cluster cells, you're defining a distance matrix, like within the cluster, right? Oh, okay, how close are they? But then in reality, this single cell is different from this single cell. Like it's like two cells. Those are two different clusters, right? So, and when you do single cell analysis, actually there's a parameter that you need to find. That you can tune. Okay, what's the resolution you want to look at? If you increase the resolution in that tool, you get more clusters. If you decrease, then you get like a smaller number of clusters. So it's kind of tricky. Yeah.
Yeah, it's sort of like you and me. In theory, technically, we're both computational biologists.
Yes.
But we both do very different things,
Exactly.
right? And so you could lump us, if you in a really low resolution way, you could say Josh and Tommy do the same thing.
Yes.
But the truth is a lot more complicated than that. In that we really do very different things.
Yeah, it's a good analogy.
That's very interesting. Yeah, that's very interesting. Well, I think it's fantastic, and I love the fact that you guys are making some progress. I think that's very exciting. And to be honest, I've got to say, I love random forests. I know people ask me all the time what my favorite ML method is, and I'm going to be honest. I love random forests because they allow you. They're easy to interpret.
Yes.
I know what they're doing. They allow all kinds of data, tabular data. Not just numeric data. You can kind of throw whatever you want at them. And I also love that you can use them as a clustering algorithm.
Right.
And I just think that's that thing right there. A lot of people were like, well, how do I cluster if I've got tabular data? Most clustering algorithms only allow numerical data.
Yes.
But random forests you can throw anything in there and it's like, I'm going to cluster this data for you. I think that's super cool.
And
so
you actually remind me of that video, that the StatQuest video. And
I know that's one of my all-time favorites. I say nobody watches it. Relatively few people watch it. And one of the big problems, the Achilles heel of this, is that that clustering is only, as far as I know, it's only implemented in the R implementation of random forest.
I see.
The Python implementation does not feature it, even though I think it's so cool. So maybe one day someone else will implement it for me because I'm not a good enough coder to make that happen. But before we go, I was wondering, Tommy, if you could give me and anyone else in the audience, if you have any advice of things that you've learned along the way. You came from basically the most humblest of beginnings, and you were all the way at the top. Mm-hmm. You went to Harvard and then beyond. And there has to be a nugget in there of something like that you learned along the way that you think could help other people.
Sure. I would like to share. So I think you need to learn meta skills, which means the skills that enables you to learn anything. So, like, just take like deep learning for example. Like I'm learning deep learning recently, and how I learn it. So first, I will read a book. Deep learning with R or deep learning with Python. There are books like that. So I will go through the examples, with code. So of course, those will not be actually related to biology. It will be like sentiment analysis, movie review, or like product recommendation. But just go through the code. It helps me to understand, okay, what parameters that I need to pay attention to, or understand the architecture of the neural network. Okay, after I finish that, I will watch your video. Or just even get a high level understanding of what's going on, right? For different architectures, convolutionally neural network. Thank you again for making those videos. And then you understand those, right? Then, because I'm working on computational biology, so I will apply what I just learned to a specific biological question. And I will use example data sets, biological data, and implement the same neural network and own that data and just get familiar with how it works. And then eventually, you need to actually do a project with what you just learned. And that's essentially how I learned bioinformatics 10 years ago, 11 years ago. So that's how I learned, because only if you can code it by yourself, or the other trick is that you want to actually share what you learned. So that's why I also write blog posts on my website, divingintogeneticsandgenomics.com. And you will see I share how I actually learned, how I actually write those neural networks. And if you can teach other people, that means you are you already well understand the problem. So that's also one trick that I use. Yeah.
And actually, you just reminded me, you do have this incredible blog, and you are currently writing posts about learning deep learning.
Correct. So I'm writing one blog post how I am learning deep learning. Yeah, it will be out I think next week sometime. Yeah.
Okay. And can you just tell us what's the name of your blog? So if anyone wants to learn deep learning and they want an additional resource, what's the name of your blog?
The name is divingintogeneticsandgenomics.com.
Divingintogeneticsandgenomics.com.
Correct.
Fantastic. Well, we should all check that out, especially if you're interested in genetics and genomics and the application of deep learning, which I think has a great potential to yield some awesome stuff. So I hope some people can check that out. Well, Tommy, I got to say thank you very much for being on my podcast. It's a real honor to have you here. I've enjoyed sort of talking with you and learning from you over the years, and so it's great to have you here. Yeah, thank you very much.
Thank you, Josh. I'm so humbled. It's my honor to be here. Yeah.
Oh, thanks.